Okay, so I'm Sharon Aviran. I'm faculty at UC Davis. I would like to thank the organizers for inviting me here. My group works at the interface between structural and functional genomics. And in particular, we specialize in the analysis of high throughput genomic data, which is obtained by a family of experiments that aim to measure certain structural properties of RNA molecules. And in that context, we work on two main challenges. The first challenge is how to maximize the extracted information out of this data. So it's a signal processing problem. And the other is once you have extracted the structural signal from the data, how can you use it to improve or to inform computational structure prediction, or generally to highlight structure-function relationship in regulatory RNA mechanisms. So today I'm going to cover work done on the first domain, which is the signal processing domain. And this is the work of my students, Mir Koleda and Pierce Radicki. And I will give a bit of a lengthy background because I think this topic was not covered before in this course, although I know that Lior Pachter gave some, like a one slide about these kind of experiments, and it's a work that I started when I did my postdoc with him. Okay, so our work is motivated by the fact that many RNAs in the cell have diverse functional roles as opposed to coding for proteins. And it's also well established that in many of these cases, their functionality derives from their structure. So structure and function are tightly linked. And therefore, it's important to try to analyze the structure in the process of characterizing the function of an RNA. Now, shown here are just three examples of families of functional RNA elements for which well-established consensus structures exist. And it's also well-established that these structures are critical to their function. But there is another thing that is common to these three examples. I'll go over them soon. What's common to them is that they also highlight the fact that RNA structure is dynamic, and it changes in response to changing cellular conditions. So the first family on the left are called RNA thermosensors. They are commonly found in bacteria, and in response to heat, they change their structure, and that propagates downstream into gene regulation. Okay, so these are the thermosensors responding to temperatures. Riboswitches is a very, very rich family in bacteria too that responds to a diverse array of ligands. They can be ions, they can be just small chemicals. Also, upon binding the ligand, it changes conformation, and that also triggers downstream events, which eventually typically regulate gene expression. So these are riboswitches, and the third are G quadruplexes, which are non-canonical, very stable structures that have been implicated in cancer and neurological disorders, and they are known to be stabilized by certain ionic conditions. For example, potassium ions are known to stabilize these structures, and they also are involved in translation regulation. Now, why am I highlighting this group? Is because for all these cases, the sequence of the RNA transcript does not change. But the cellular conditions do induce change in structure. And this is something that if you use sequence information alone, you won't be able to get this information. This is where experiments become very, very, very informative and useful. And this is only one example where experiments are needed. Okay? Now, suppose I want to predict RNA structure computationally. I'm sure many of you have learned about that in classes, bioinformatics classes. The most popular approach is called NNTM, standing for Nearest Neighbor Thermodynamic Model. Basically, a very rich model of the energetics of folding for RNAs. And the input is the sequence, the RNA sequence information. Then incorporated with thermodynamics consideration, there are two most common outputs of these kind of algorithms, and they're very popular. One is a single structure called the minimum free energy structure, so it's the most likely or most stable structure out of many, many possible. The other type of outputs is richer in the sense that you get a subset, say, of 1,000 or 10,000 suboptimal structures. The second approach is considered more accurate because it does give you a better view of the energy landscapes, but the downside of it is that it's more computationally expensive. So most people will go for MFE, and people who are more into RNA structure often will try to go with the what's called the Bonds ensemble approach. Now, the main drawbacks of these algorithms, as popular as they are, is that their prediction accuracy is not so great, and it also known to drop pretty quickly with increasing length of the RNA. So RNAs that are a few hundred nucleotide long already reach accuracies of like 60, 70%. They're also known to be performing poorly when you are trying to predict structures inside a cellular environment, because the energy model here was made based on experimental measurements in vitro. And these often don't carry over very well to in vivo, like to the real complexity of the cellular environment. So these are the main limitations. Another limitation, if we want to work on transcriptome level, which is something that we can do in recent years, these do not scale well. Although in one RNA at a time they work pretty quickly, I'll show you later, they don't work well at transcriptome level. Okay. So now, another way to probe structure is by what's called collectively structure probing experiments. It's a family of different techniques. It's a classical approach. It's not new. It's from the end of the 70s. And here I'll demonstrate it on one particular probe called SHAPE. SHAPE is just an acronym for selective 2'-hydroxyl acylation analyzed by power extension. It's the most popular probe today, but there are many others. And generally, these experiments give you a snapshot, an average picture of all the RNA structures in the solution that you are probing. Their goal is to discriminate in many different ways between paired and unpaired nucleotides. And the main limitation is that they cannot really tell you the entire structure. And when I talk about secondary structure, I mean who pairs with who and who is unpaired. They cannot tell you that. They can just tell you something about whether each nucleotide is paired or unpaired without telling you who is the partner. Okay? So it's a kind of partial information, and therefore typically they are not used alone, but in combination with something else. Now, SHAPE itself targets the 2'-hydroxyl group at the backbone of each nucleotide. So all nucleotides look the same in terms of their backbone. And it's based on the observation that if in unstructured or unpaired regions of the RNA, the backbone is much more flexible to move and therefore more readily reacts with the SHAPE reagent. Whereas in structured or paired regions, it is less reactive. Sometimes it is, but much less. Okay? So the discrimination between paired and unpaired comes from the extent of reactivity of each nucleotide to the SHAPE reagent. Now, this reactivity results in a modification of the nucleotide, and ideally, we will find many more modifications in the unpaired regions versus the paired ones. So what SHAPE really measures, and again, that varies between different experiments, the SHAPE experiment really measures backbone flexibility, which has been shown to correlate very well with nucleotide base pairing state. So it's an indirect measurement of a base pairing. So as I said, this is a classical approach, and now I'll show you the old style of SHAPE experiments as they were until 2011. So basically, you take your RNA. This is all done in vitro. At that point in time, you fold it. Then you put the SHAPE reagent in, and it reacts preferentially with the unpaired nucleotides at the various molecules. And then you anneal a primer that is complementary to the 3' end of the RNA. And then you throw in a reverse transcriptase. Reverse transcriptase basically naturally just will synthesize the complementary DNA to the RNA. But when it hits the modification location, it cannot traverse. So it stops there. So what you end up with is a bunch of fragments that are subsequences of your RNA sequence. And if you dissociate them from the RNA and you sequence them, and you count how many you had of each type, you will get a good sense of how extensively each nucleotide was modified, which should indicate its pairing state. Okay, so that's how it works. Now, since 2011, we had a lot of things happening in the field. So the newest version looks like that. Basically, instead of a single primer, you have random primers, very similar to RNA-seq. And random primers basically bind everywhere. And modifications happen as before. You can do it at the entire cell level. And reverse transcription will stop at the location of modifications, right? And then you'll get fragments, and you'll map it to the RNA transcript, so you'll see where everything stopped, and you can count stops. Now, it's not as simple as I described. Often, we also add a control experiment where you do exactly the same without the modification to control for noise, because there are stops that are not modification-induced. There are also versions where the reverse transcriptors will follow through but will introduce a mutation. So we have a variety of ways to detect those modifications. But generally, that's how it looks like. Then you count fragments here, you count fragments here. There is a whole informatics box in between as to how to combine the counts into the final output, which are called nucleotide reactivities. So you get this bar, you get this signal. And high reactivity indicates high likelihood to be unpaired. Low reactivity indicates high likelihood to be paired. That's basically how it looks like. Now let me just try to convince you with a simple example that there is information in this data, and also that this data is not perfect. So here is a known secondary structure of a regulatory bacterial motif. We can see here 3 hairpins. Now, the color coding is according to the magnitude of reactivities. So red is very high, black is no reactivity. And what you see here, this piece, these bars, correspond to the free stretch on the left. Okay? So that's an unpaired region, and you can see reactivities are fairly high. Right? Now we'll go into this hairpin. So this is that hairpin. So how does it look like? First, you have a stem. So you have very low reactivities. Then you have a loop. Reactivities go up. Then you close the stem. Reactivities go down. Okay? So there is something in there. Now let's look at the next free stretch. It's reactive, but not as reactive as the first one. Okay? So not perfect or not deterministic. The other hairpin, again, stem, loop, stem. Another free region, and stem, loop, stem. Okay? So what you can see here, that you can see the structure if you have enough experience, but it's also not perfect. The hairpins don't look the same. And that's why typically you do need to get input from another method, typically a computational structure prediction algorithm. And even if this looks pretty obvious to you that these are 3 hairpins, when the RNA gets longer, there's no way you can figure out the structure from that. Okay? Now, in the last 6-7 years, we have seen rapid growth in this area, and today we have the following capabilities. First of all, these experiments are transcriptome-wide, so a single experiment can probe most of the structures in a cell population. So it's called the structure one. The second is that we have multiple probes. It's not only shape. Those probes are complementary to each other. They probe different local properties. And sometimes people run several experiments in parallel. Even for a given probe, there are many, many ways to do the experiments to extract the modification locations. So that also results in very different data types. Okay? And we can, as I said before, we can do it in diverse conditions. It's not only in vitro and in vivo, but also different temperatures and with different ligand concentrations, which all these together affect the types of the data that you get at the end, or the readouts and their statistics. So if we want to take advantage of these new data sets, we want fast, flexible, and data-driven methods to predict or analyze structure. Fast because we are at transcriptome scale for the first time. Flexible because we have many, many different data sets generated by different experiments. So we want an algorithm that quickly adapts to the data type rather than having to train it and characterize it before you use it. And data-driven is, ideally, we would like to exploit the information in the data as much as possible. especially in scenarios where the thermodynamics paradigm is not very accurate. So just to give you a sense of when I talk about multiple techniques, this is a new review from last Friday. These are just the number of techniques currently out there. And every few months there is a new one. And all these techniques apply for any given probe. So you can do it with any given probe and any given condition. So you get a lot of different data sets at the end. And generally, this review does cover the latest and greatest in the field. So if you want to know more, I recommend taking a look. Now, suppose I want to use it to predict structure. So typically, you would face two main challenges when you try to do that. The first challenge is bridging between different resolutions. The resolution of information here is at the nucleotide level. The resolution of functional elements is typically at least a few nucleotides and often tens of nucleotides. Functional elements don't function at single nucleotide level. At least 5, 6, and often many more. So going from here to here, especially when the RNA is long, is not trivial. The other thing, as I mentioned, this is partial information because that does not indicate who is paired to who. So these are the major challenges in using this data to predict structure. And the standard approach that has been very popular in the last 10 years is called data-directed secondary structure prediction. Basically, you take the NNTM paradigm, which is based on dynamic programming algorithms, and you find a way to translate the data into the domain of the free energy, and you combine them together, and you get the usual output, the MFE or the Boltzmann ensemble. That has shown to generally improve accuracy fairly well. It varies between, on a case-by-case basis, but sometimes you get 20, 30% improvement in accuracy. But the approach still suffers the same limitation of the dynamic programming paradigm because this is still the computational bottleneck. So it's not scalable to transcriptome level. So this is not something you can do to fold an entire transcriptome. Okay? So these are the opportunities and the limitations. And that motivated us to develop Paterna. So what is Paterna trying to do? Paterna gets an input of a data set, like a lot of RNAs with their structural profiles. And it also asks the user, what are you looking for? So the user can say, well, I'm looking for that structural element. Okay? And there is a way to encode it into a way Paterna understands. And basically, Paterna takes the desired motif and scans the entire data set and site by site. And at the end, gives you a list of sites according to how likely they are to harbor the motif. Okay? And that is done by probabilistic modeling and calculation of the probability of the motif being there given the data. Okay? So it's a probabilistic framework. It has two advantages. First of all, it does not impose any thermodynamics considerations. So you can do it in experimental conditions where thermodynamics is not very applicable. And the second thing, it's also unsupervised, so it doesn't need to be trained on a set of structure profiles that have known secondary structure, which has been the case before for all other algorithms. Now, what is it based on? It's based on the following observation. So here are the three hairpins that we just saw. And as I said, the magnitude of the signal is different between each of them, right? It's not perfect, but they all have the same shape. The same shape, basically, all hairpins look like a valley, peak valley, right? So there is a characteristic signature to the motif, okay? But it's not deterministic. So that leads to statistical characterization of the patterns that arise from different motifs. So this is what Paterna is doing. So Paterna is just a pattern recognition algorithm inspired by speech recognition engines. And the first thing, it uses a simplified representation of what structure is. So structure is commonly, at least secondary structure, is commonly presented as who pairs with who and who is unpaired, so base pairing pattern. But Paterna doesn't do that because Paterna can't extract this information from the data anyway, right? So Paterna simplifies it into a linear representation of the RNA as paired, unpaired, who is unpaired, right? Because that's the best that we can do. Now, just keep in mind that paired unpaired is really a simplification, because shape often will reflect also interaction of the RNA with the ligand and so on. So paired unpaired really stands for reactive unreactive, but for simplicity, let's think about it as paired unpaired. Okay? Now, as we said, the rationale is that we can't really get more. Now, by doing it, we compromise accuracy. Why do we compromise accuracy? So here is an example of a paired unpaired sequence, and all these three structural motifs apply, right? They all map to the same sequence. So you lose some ability to discriminate between certain motifs. So what we do to alleviate this problem is we can also impose sequence constraints. What do I mean by that? So for example, if I know I'm looking for this hairpin, I can look at the sequence and say, are these pairs feasible in terms of who pairs with who, right? If they're not, it's out of consideration. And that helps improve the FDR and accuracy in general. Now, there is one really important thing about this representation, is that this is what confers paternity speed. If we had gone to the old representation of who pairs with who, which is how the dynamic programming paradigm works, we would not be able to do that in a few hours, as I'll show you before, but rather in a few months. So this is what gives paternity speed at the compromise of certain limitations. Now, Paterna has a statistical model at its core, and it has two components. The first component are basically two distributions. It's the statistical values of reactivities that you expect to see from unpaired nucleotide versus the reactivities that you expect to see statistically from paired nucleotides. If a probe is good at discriminating between them, generally you would expect the unpaired to be higher than the paired, right? But they do overlap. It's not perfect. So we're trying to figure out those distributions. And the second thing we're trying to figure out is the general, oops, why is that? Shall I try, let me try it again. Okay. All right. Now, the second thing we're trying to learn are the general characteristics of the structures in the solution, perhaps those of the entire cell. So now, what characteristics are we looking into? Structures have the property that paired nucleotides come in runs and unpaired nucleotides come in runs. So that can be captured in a first-order approximation by a simple Markov model, switching between paired and unpaired. So, and for example, if you tend to have long stems, you tend to stay more in the paired position until you switch to the unpaired. So this is the second thing we learn. We learn the distributions and we learn the general properties of structure. Then we merge them together. So for this part, we use a Gaussian mixture model, right, to just give flexibility in describing the empirical distribution. This is just an HMM. Together, they form GMM-HMM framework. It's a very well-studied framework and has been applied very successfully to speech recognition since the end of the 80s. So the whole methodology is already developed for this. So when you take them together, this is how Paterna looks like. It has a training phase on the top, and after training is done, we do the scoring. So for the training, you don't even need to know what the user is looking for. You just need to have all the structure profiles, and you do unsupervised learning by an EM algorithm or a Baum-Welsh algorithm, basically maximizing the likelihood function. So you are looking for the model parameters, which are the distributions and the transition probabilities, right, that maximize the likelihood of the data. So the E step basically calculates probabilistic assignments for the hidden variables. What are the hidden variables here? Are you paired or unpaired? So this is the state of each nucleotide. So we calculated the E step posterior probabilities for each nucleotide, given the data and given the current model parameters. We do it by a forward-backward algorithm. It's very, very standard methodology. Now, after we have calculated the posteriors for all the nucleotides, so we know the hidden variable assignments, we go and recalculate the Gaussian mixtures and the transition probabilities, okay, given the current probabilistic assignments, okay? And so we iterate on and on and on. That's a classical EM algorithm. After we have converged to a maximum, we take the trained model, meaning we work with two distributions and a transition matrix, and we start scoring the entire data set. Basically, then the user says, I want this motif. So the motif corresponds to a path on the trellis of paired unpaired, right? And basically, every location in the transcriptome is scored for the probability of having this path plus having the observable, which are the reactivity, okay? Again, same building blocks as the forward-backward algorithm. These are all probabilistic calculations of either a single nucleotide probability to be paired or unpaired, or a whole path to be a sequence of paired unpaired. So that's all not new, this is all standard. Okay, so now to validate if this works, we did a few things with increasing level of complexity. So the first thing, we took a data set that was produced by the lab that produces the best shape data, is the lab of the guy who developed shape, Kevin Weeks. And they profiled like 20-something RNAs, which all had reference structures, either by crystallography or by phylogenetic analysis. So there was a reference or some kind of ground truth about who is paired and who is unpaired, and there is a shape profile for each RNA. So we use the ground truth as what's really out there. And then we ran Paterna and looked at what the crystal structure is saying. So the first thing you see here on the left are just the two distributions. These are Paterna scores for the sites where really the search motif was versus those where it wasn't, right? So the first thing is you see enrichment. You see that scores are generally higher when it's there. So that's a preliminary validation that at least we're doing something good. And the next thing was to look for a bunch of motifs, so many hairpins with different lengths and many hairpins with bulges and different things like that. And again, looking at the reference structure as the ground truth and saying, did I get it right or not? And here what you see is we compared Paterna. So Paterna is in blue to MFE, the single structure, and ensemble. So ensemble is on top and ensemble is better. Now MFE and ensemble can be directed by the data or not directed. And you see that Paterna is kind of in the middle between them. Now, clearly, ensemble prediction outperforms Paterna and MFE. It's not surprising. And moreover, all these structures were probed in vitro. So the assumptions of the model also are more applicable. But the downside of ensemble is how much time it takes. So to give you a quick idea, if you want to, say, fold a transcriptome or detect a motif in a transcriptome with Paterna, Paterna will take several hours. If you want MFE, it can take 3 to 5 weeks. And if you want ensemble, we're talking about the order of a year. Okay? So you compromise the accuracy for speed. Okay. So now the next validation was going for some more complicated system. So what you see here is a new experimental system called co-transcriptional shape-seq. So shape-seq is just the type of probing I discussed. Co-transcriptional means being able to capture the intermediate folds of transcript as a transcript is being transcribed. And we do know that things fold as the transcript is elongated, right? And they also change the folds. So the fold that you see at the end of the complete transcript is not what it was in intermediate steps. And that actually matters a lot for riboswitches because they do take different folding pathways in the presence or absence of their ligand. So what you see here is just how they did it. They basically embedded stops at any point in the transcription, and then they captured all the elongating transcripts with their structures by shape. So that's what they did. And their data looks like that. So you see those matrices. Every line here is an intermediate transcript. So as we go down, the transcripts get longer and longer. This is the riboswitch that was probed, and these are the known final folds after transcription, okay? So with the ligand, it converges to this fold. Without the ligand, it converges to that fold. And what you can see here is the co-transcriptional reactivities with the ligand and without the ligand. Now, what they did here, this is an experimental group. They looked at the differences between the matrices, which you can see here, and they just tracked single nucleotides that seemed to change. They also looked at the literature and to what's known and unknown about the folding pathway, and they came up qualitatively with a model for how bifurcation happens, like when at a certain point the presence of the ligands biases the pathway into one conformation versus the other. Now, I happen to know the first author of this paper very well because we collaborate with this group, and he told me that he stared at these data for weeks, okay, in order to figure out what's going on. So we decided that because paternal works at the motif level and not the nucleotide level, how about we try to automate it? Can we find the entire feature in the data instead of going nucleotide by nucleotide? So these are, again, the final conformation without ligand. And what you see here is also matrices like the one before, where when we go down, transcripts become longer and longer. And the heat map here are paternal scores. So, and in this search, we're searching for this pseudonode. Okay, so this is the pseudonode that we're searching, and we expect to find it in the presence of the ligand and not in the absence. Now, dark scores means that we see it there. The data supports this motif. Light means it's not there. Okay, so what you can see is that the pseudonode was in both conditions in the beginning. And here you start seeing the bifurcation. Without the ligand, at some point it's gone, almost gone, right? With the ligand, it persists. There is also some noise in the data, but it persists through the end. Now, what you can also not see here is that those pixels are much darker than that. So we also see the pseudonode here in higher abundance, okay? But that's hard to see visually. Okay, the third application was going transcriptome-wide, and this time with a different technique. This technique is called parse. It came from Howard Chang's lab in Stanford, and it operates slightly differently, but the general idea is the same. The main difference, though, is that here, if a nucleotide is paired, you will expect positive scores. It's a bit counterintuitive. And if it's unpaired, you would expect negative yellow scores, okay? So already shows you that the data type is different and the behavior is different, okay? So we trained paternal and parse data on that specific data set. And what I show you is just one small example. We were looking for an element called a ribosnitch. What is a ribosnitch? A ribosnitch is a case where a SNP or a SNV introduces change in the structure, which often is translated into functional consequences, okay? So the SNP location here is in red, right? And these are the two predicted structures for the two alleles predicted by the authors of that paper. So we said, okay, let's look for them. Now, we had a family trio. It's a mom, dad, and a child. And in this case, dad is homozygous for G allele, mom is for A, and child is heterozygous. So, okay? So, and now, how do you read this? Basically, the colors here indicate the ideal pattern, the pattern of the thought motif, right? And what you see, the bars are the real data, okay? So that is homozygous for G. Now, you can see that when we look in the dad's data for the A motif, right? So the wrong motif, over like 2 million sites, it's ranked at like 5%, okay? If you look for the dad's predicted motif in the dad's data, it's ranked above 0.01%, so ranked much higher. But it's not the top one, because there are other sites in the transcriptome that even better support that motif. But you can see a discrimination between the wrong and the right motif. We also did some spiked in perfect information structures, but I'm not sure how much time I have left. So anybody knows how much time I have? 7 minutes. Okay. So I'll continue. All right. So a little bit about the model accuracy. As I said, we do unsupervised learning. So this is just an example of how it looks like. So you are trying to guess two distributions, paired, unpaired. One of them should be lower generally than the other. And you start by just spreading the Gaussian components from left to right. And this is an extreme example. That's not really how we do it. Here we have three components, three blue, three red. So blue will be for unpaired, red will be for paired. And the empirical distribution of all reactivities together, because you don't know who is paired or unpaired, is in gray, right? And you want to fit it, okay? So this is basically how it behaves from iteration to iteration. So you can see that it basically slowly learns the distributions, right? So the fits become better, okay? So that's basically how it looks like. But again, it's a bit of an extreme example. Now, in order to see if it made sense, we got back to the data set from Kevin Wick's lab because we had the ground truth. We had reference structures. So what we did there, we looked at the reference structures and we classified the nucleotide according to whether they are paired or unpaired in the ground truth. And then we got to empirical distributions, the gray ones. And what you can see here are the paterna fits. So the Gaussian components are in dashed lines, and the overall distribution is in a solid line. Overall, it fits well, but we do need to do a better job at the tails. So we're still working on it, but you can see the difference between supervised and unsupervised learning of these distributions. Now, a little bit about benchmarks. As I said, if you go transcriptome-wide, paterna will typically take on the orders of hours. As you can see example for the parse data set and for simulated data sets. And what you can see here are benchmarks between paterna and predicting MFE and predicting ensemble. So you can see that ensemble for a transcriptome, a typical transcriptome, can take over a year to do. And even MFE would take the order of a month. So this is the main advantage of paterna. Now, to conclude, let me mention a few applications that we thought of, which is why we designed this algorithm. So first of all, as we said, you give me a desired motif, and I will just highlight candidate regions across the entire transcriptome where it can be. But we also, like many genome-wide screens, we also have a lot of false detections, right? So one thing we could do is actually use this in conjunction with the thermodynamics approach. You can use paterna to narrow down the search set, and then on a smaller set, run thermodynamics considerations that can be data directed. Another thing you could do is there are many labs that are interested in general characteristics of structures. So for example, they want to know if UTRs are more structured than coding regions, which is what we think it is, but there is no direct evidence of it. So you can, for example, see are there more hairpins generally in UTRs versus coding regions. So you can do that kind of searches. Another thing that I didn't show you, which is very important in structural biology, is that paterna can highlight shifts in ensemble composition. So I showed you the ensemble story in the context of riboswitch, and that's very, very important for function, because the ensemble composition between competing conformations is what determines the functionality of an RNA, and also when you design new RNAs, it affects their efficacy. So it's very important. So paterna is able to really show you, say you look between two different conformations, it can definitely show you if the ensemble shifted towards one or the other. It cannot show you how much there is of each one. That it cannot do. As I showed you, you can also try to understand folding pathways using new experimental systems such as co-transcriptional shape-seq. And for example, if you design drugs that bind RNA, which is a very hot topic now in industry, there are many new companies who either design RNAs drugs or target RNAs with small molecules. You can profile your transcripts for target and off-target sites if you know that your molecule binds certain motifs. Okay? So these are just a bunch of applications. So to summarize, paterna basically does not work with thermodynamic considerations, which is good in certain scenarios. and also doesn't need any training, reference training set, which makes it easy if you work with a probe that was not heavily characterized, such as shape, then you won't have this kind of information. So you want to do unsupervised learning. It's compatible with many different techniques because it learns the property of your own data set in the conditions, whatever you did, and not carrying over from other people's work. It does characterize for the first time structural information at the motif level, as opposed to nucleotide level directly from the data. And on the limitation side, in addition to the limitations I already showed, one big limitation is that you need to know what you look for. And sometimes, for example, you're looking for thermosensors, but not in bacteria. In plants, they may not look exactly like the ones in plant. So you may need to think of many variations and so on. So it's a bit limiting. And the other issue, which is still an issue for this type of experiments, and is not an issue very much with RNA-seq, is that you do need sufficient amount of data of high quality to train. And those experiments are still not totally there. They do have some well-covered transcript, but a lot of it is still poor quality. And that's about it. It's available on GitHub. And Mir Koleda is the one who designed the initial implementation of Paterna. And Piers Radeke is now working on improvement. Was funded by NIH. And thank you for your attention.
So first, I would expect that for more complex patterns, where you have more information, your ability to distinguish will be higher. Can you quantify that?
We're actually working on that right now.
Okay.
Yeah, and you're right, generally. Yeah, we're working on better characterizing it.
Quantify how much information you have in every pattern.
Yeah. Generally, longer motif or more elaborate motif, you will have a better discrimination power. Yeah, absolutely.
The other thing is that the ensemble is the Hofacker group of Vienna?
No. So in the RNA world, there are like 2 or 3 big players. There is RNA.
Hofacker
is? He is.
Okay.
He's the big.
Oh, okay.
So there is Dave Matthews, who actually is the one who developed the data-directed structure prediction algorithm. He has RNA structure. And there is Hofacker. There is another Japanese group, but they are smaller. So there is Hofacker from the Vienna RNA. What these people do is that whenever there is a new method or a new algorithm, it's within a year or 2, they all implement it. So it doesn't really, even if your competitor is the one who came up with it, they make sure they're pretty similar to each other. Exactly. It's a very friendly competition there.
Ensemble is the Wellcome Trust, no? So, and
No, it's not that ensemble. So it's just, so it's called the Boltzmann ensemble. It comes from biophysics. It's basically, it's a statistical distribution on all possible conformations. There are a ton of them. Now you can't really get all of them. So you can either theoretically, under the assumptions of the model, calculate a partition function, which is the normalizing factor, and therefore you can transform the energies into probabilities. Or what's more common is to get a sample, a statistical sample that is a realization of the ensemble, but you won't sample typically more than 10,000 experiments because there is a visualization problem, right? So of course it doesn't capture the whole ensemble, but the idea is that it will sample the most frequently occurring suboptimal structures. And it has been shown in many ways that if you take samples of the ensemble versus MMP, you would get more accurate results because those models all suffer from inaccuracies, right? There are simplifying assumptions there. And they have a hard time distinguishing between structures that sometimes are not similar at all, but have very close free energy. So the ensemble approach says, okay, we're not choosing one winner, we're looking at the top, not top, like the most frequently observed 10,000, and let's look at the average or the frequency of this helix here and there and so on. So it's a more informative and I think a wiser approach. And it depends, typically, if the user is more familiar with RNA structure prediction and limitations, we'll go for the ensemble. If the user doesn't know anything, they will go for MFE because it's a simple answer. It's generally, that's what happens. Yeah.
So if I remember correctly, if you look at the loop, the reactivity is not uniform, like in a loop. So it's like higher, lower, depending on the method in the beginning and in the end, and higher in the like center nucleotides in the loop. How do you take this into account? Maybe have like some like additional states in your
So generally, it will be higher in the middle of the loop, although there are exceptions to that too, right? Because there are other factors weighing in there, like tertiary interactions that I didn't talk about. But it's true that you can extend the context. You don't have to look only on your previous nucleotide, right? Maybe you can look at the right at the left, or two to the right, two to the left. It's something we're planning to do, but we haven't done that yet. I think it's more than that. It's more than that. I think it has to do with the actual sequence that is there, and also the stem that comes after it. So I think it's not that trivial to capture, but definitely extending the context may help. We don't know how much it will help, but we're planning to do that. What I can tell you, though, is that in terms of correlation between reactivities, reactivity and its neighboring reactivities are pretty well correlated. It's around 0.3. Reactivity and its second neighbor are almost not correlated, right? So in that sense, it doesn't buy you much to extend the context, but you're right that it might capture patterns that are more sophisticated than what we can currently do. All right, thank you.